welcome, Gosia, to the live Q&A session of your seminar. Really appreciate you joining us today. In particular, thank you for taking the time out from your lab's Christmas party to join us today.
Thank you. Thanks for inviting. Pleasure to be here.
Great. So it's just a reminder for those watching that please take this opportunity to ask any questions you may have by typing them into the question box, as well as please vote for different questions, or if there's a question that you would particularly like to see asked, please do so, and we look at those rankings when we select questions for this Q&A session. So I'll just start off with a first question, Gosia, that's right at the top of the list. So you obviously have this leadership role in open targets. And so the question is, does working within open targets consortium influence your thinking on the scale of experiments needed? And I guess maybe you should just briefly introduce what open targets is as well.
Sure. Yes, I really like this question because the answer is yes. Indeed, let's introduce what open targets is. It's a consortium here at the Wellcome Genome Campus that brings together the Sanger Institute, EBI, and 4 different industry partners. That's GSK, Sanofi, BMS, and Takeda. And our overarching goal is to use genetics to inform drug identification, drug prioritization. And we work by building together projects between the faculty at the 2 institutes and our partners in the industry. And those projects really aim to think about how do we leverage genetics, how do we build projects that can utilize different genomic approaches to better inform drug discovery. And we're quite well known for our Open Targets Genetics platform, which really it's a website it's the output of all our work. So behind the doors, we put together all sorts of different sources of information from clinical trials, from genetic studies, from gene expression. Really, it's a comprehensive, integrated resource of different sources of information that builds support towards a role of certain gene or pathway in disease biology. And in addition to very strong informatics arm of Open Targets, we also have experimental program, and within our experimental program, we specifically generate data, and that data also feeds into the Open Targets platform. So the question is, if by working in the Open Targets Consortium, does that influence how we think about the scale of experiments needed? And absolutely, yes, it helps us shape projects that are large scale and that are comprehensive. And I think looking at the research in my own group, where it really helps is it brings focus to our projects. So it's no longer often what we like to do in academia, just follow our curiosity and have projects that are perhaps pure basic research and just out of curiosity. We actually often have to think about if that particular project is going to generate a list of targets. And that's actually conceptually slightly different than just thinking about how do I build an interesting project, where does my curiosity drive me. So I think that's a significant difference that I definitely recognize in myself and how we approach thinking about new projects. But equally, when we analyze the data, we also think about how does that help us to get us closer to the targets? How does it allow us to prioritize new targets, and what can we do with it next?
So can I just follow up a bit on that? Because you talk about the importance of scale in those projects and being kind of unbiased and comprehensive. And then you've talked a bit about focus. And some people might see those as almost opposite things, because people focus to narrow the scope and narrow the scale. So, I mean, I wanted to pick that apart a little bit. What it is, presumably then it's something about, the statistical power that you can leverage by the large scale data sets in very focused questions. Am I correct in kind of assuming that? And why is that maybe different than the way you would have done something in a more traditional academic type setting where you're kind of exploring new aspects of biology?
Yes. So let me fall back on some of the projects I was describing here. I spoke about Cheer's method and our results of identifying SNP enrichment for immune disease variants in activated memory T cells. That project really originated with this broad question, which was like, we knew there's a signal of GWAS-associated variants in T cells, and we wanted to explore that comprehensively across the different conditions that we anticipated were relevant for developing immune diseases. So there's that breadth with quite a focus. We focused on 3 different cell types in that case. And the question was quite fundamental, but it was focused. Our goal was to understand, are there specific cell states which will get us closer to understanding what is the disease pathobiology through which disease-associated variants manifest. And we landed at these results of activation of memory T cells. And actually, initially, where we thought we would take the project was that we would perhaps use different cytokines to generate EQTL maps of T cells in response to different cytokines. But having seen those results, we actually sat down and thought, will that be the most informative way to get us closer to target identification? And that's where we thought, actually, we need a more specific focus at this point. And we thought that most impactful subsequent approach would be to look at T cell activation trajectory and use the technology that was very promising to give us single cell resolution, or resolution so detailed that we didn't have before. So there's this fine balance, as you say. We had that breadth, and then once we had a good idea that this is disease relevant context, we were a little bit more focused in that context. Does that answer your question?
Yeah, no, that's very interesting. It's very interesting to hear the evolution of the thought process, both from the design to actually the implementation, and then going very deep in the memories T cells.
And just I'll build on that. And I think where perhaps also where I see a big influence of open targets on thinking for us about this project is now that we have this list of EQTLs and potential list of targets, we're now thinking about how do we validate that, right? Like, what does it now mean to validate those genes, and what's the context that we need, and how do we convince ourselves that those EQTLs that we're identifying are truly meaningful to the disease biology? And and I think one important example here how working with open targets helps shape this project is, for instance, we think about taking forward genes that are tractable, right? So we're not only taking a list of genes that come from our EQTL study, but now we're actually plugging in information that comes from our interactions with industry partners, that come from the Open Targets platform, which help us to say, well, take those genes because they are more likely to be successful drug targets in your subsequent experiments. And I think that's something that I identify as something very different to how we thought about our experiments, say, 5 years ago in the group.
Yeah, no, that's very interesting. Good. I think we'll maybe move on to a question from Roser.
Yeah. So your talk and your work is focused on immune cells. And yeah, someone wonders if the same principles will apply when interpreting causal variants for other diseases that are not immune related.
Yeah, absolutely. So this type of approaches are not only for immune diseases. In fact, I would say for other common complex diseases, similar approaches might almost be easier, especially if you have good understanding of the disease causal tissues. So the problem with immune system is that it's very dynamic, and there are really many cells that are engaged, and they interact with different tissues. So it really is very complex. But if you think about a disease such as, for example, type 2 diabetes, you have a much better idea of what's the causal tissue. And actually that saves you a lot of that initial work to identify the right cellular context in which you should be studying disease variants. And so absolutely, this kind of approaches are applicable to a whole range of different complex diseases. And if the disease has a better tissue signature, then you're actually even more likely to go into deeper functional follow-up studies.
Yeah, thank you.
So I'll ask a question now. Somebody's asked a question, what are the barriers? Are they technological, experimental, computational barriers to generating single-cell EQTL maps for the analysis of disease-associated loci?
There's quite a few. I mean, this technology is advancing really rapidly, but of course, one of the barriers still are the costs. Having said that, I think there's really tremendous momentum right now towards generating single cell EQTL maps across tissues and across states. And we already see such data sets emerging across different tissues, across different cell types, and across different cell states. In terms of challenges, I mean, one obvious challenge is mapping those EQTLs because of the size of the data sets. You need to be clever about how you're analyzing this data in an informative way, because you're really dealing with a massive amount of data set. And I think we're still developing new models that can be applied to such analysis, because you get much more information from single cell data sets. You can not only look at single cell resolution, you can start looking at the trajectories of how cells differentiate and evolve within different conditions. So there's a lot of opportunity in terms of building new statistical models that will be able to perform very interesting new types of analysis. And of course, in terms of the technologies also, there's still some challenges in the sense that you need to have certain coverage of cells to have sufficient number of cells to accurately estimate gene expression values. And that has to do purely with the nature of the technology, high dropouts in terms of capturing gene expression and so on. Yeah.
Yeah, so quite a lot there. Yes, thank you. Mathew.
Great, thanks, Gosia. So it's a specific question, but I think I might ask it in a more general way, or at least I'll ask you to kind of extrapolate more generally. So the question is what stimulation was used for your dynamic EQTL mapping? And have you observed differences using different stimuli in terms of cell states and gene sets and dynamics? And I guess generally the thing that struck me is there's so many variables here in terms of this model system, the stimulus, the intensity of that stimulus, the duration of that stimulus. So I wondered if you could kind of comment on, I guess, the specific question, but more broadly, what you feel the opportunity is, or the challenges, or how we overcome this huge combinatorial problem.
Well, you really captured my dream project over there. Having the ability to generate this data set where you can manipulate the amount of activation, because I mean, in essence, that's what our cells are seeing in our body. They're not seeing exactly the same amount of stimulus, right? They're responding to variable amounts of different
and multiple cues simultaneously as well, right?
Absolutely.
Yeah.
Yes, 100%. So
Yeah.
of course, in here, we're simplifying the model. We've used standard activation with beats through activating TCR and CD28. It's a broadly used stimulus, but it's also a very strong one and probably physiologically not that relevant, although it's very widely accepted. Everybody uses beats. And it gives us a good idea about the dynamics of cell response. But as you've really very well captured there, you know, in an inflammatory condition, the cell is responding to ligand receptor interactions, and Roser will know much better about that. There are cytokines in the environment that cell is reading, and all of those are modulating the signal. And then you'll have the variable levels of the stimulus itself. So there are just so many variables in there. And I'd love to one day be able to do that kind of experiment, but you know, as you say, combinatorics of that, they just go exponential. You need to simplify the model a little bit, but maybe I'll spin it out a little bit and say, I think we're in the next years, we'll see a lot of fascinating data emerge is from EQTL studies, single cell EQTL studies from patients' tissue. So you'll be actually, we will start to be able to look at those individual cells, what do they express, and start linking how genetic variation modulate your antigen presenting cell and on all the co-stimulatory molecules on the antigen presenting cell. And then at the same time, in that inflamed condition, what happens to the T cell and how does the T cell respond to all of that signal present, and what's the role of genetics in controlling the interplay between those two cell types. So I'm a big fan of doing experiments in a dish as well, because it gives you the environment that you can control, of course. But I think there will be a way that we will be able to get clues from the tissue, from the patient samples, and then be better informed about what kind of experiments we're going to do in the dish, because otherwise it will be very challenging.
Great. Yeah, thank you. It's very interesting. Lots of work to be done.
Oh, yeah.
Yeah, so there is another question about EQTLs in general. Why do some dynamic EQTLs have linear effects where others are not linear? And if this is telling something about the gene regulation?
Mm-hmm. So I think it definitely tells us something about gene regulation. And I showed as an example some of the well-known gene markers of T cell activation that we see, that some markers come like in the late activation, others come in the early activation. So there is dynamics of gene expression as such. So I hope we are capturing some of those effects, and we're seeing what's the genetic regulation effect on the gene expression for such genes. Another concept here is perhaps that some of those genes may actually have EQTLs sort of in the middle of the activation trajectory. And if you have an early activation time point, say midpoint and late point, and you see EQTL in the midpoint, in the linear context, we wouldn't be able to capture that, but that would come out as a dynamic EQTL. But in fact, it will be a little bit more of a time point specific EQTL really. But again, that's a reflection of gene expression regulation.
Yeah, thank you.
We're coming close to the end of the Q&A session, so I'm going to hand over to Roser to close.
Yeah, so, well, thank you so much, Gosia, again, for your presentation and joining us for the Q&A session, and also the audience who joined the virtual session. This is our last Sanger seminar session for 2020, and we are delighted to confirm that we will continue these seminars for the next 6 months. So please look out for announcements. Next month, a speaker will be Dr. Jeff Barrett on the 20th of January. So Jeff has returned to Sanger recently to lead the Institute COVID-19 efforts. And Jeff will describe the establishment of the Sanger COVID-19 pipeline and the analysis of COVID genomes. Also, I would like to make you aware that these seminars and the questions and answer sessions are active on this webpage for futures viewing. We have been delighted that this series has allowed us to share Sanger science with a diverse scientific audience around the world. Thank you for joining, and we look forward to seeing you next month.